Okay, so can I ask you what this key is here? What
Oh yeah, this key, yeah, that's a key that when I was an undergrad in Trinity College, my third year I joined the subject of genetics, and there was a building for genetics at the time, a really old tumbling down building that I think was originally Oscar Wilde's dad's hospital for doing operations in. So it was actually quite
Was Oscar Wilde's dad a surgeon?
Yeah, he was a big Sir William Wilde.
God,
he was
I
a
didn't
big
know that.
eye surgeon and stuff. So, but anyway, so the building's been knocked down since and replaced with a much more flash institute. But at the time when I joined it, there was a guy, George Dawson, who was the head of the department. And the very first thing when we were just this kind of little green third year coming into the next department, he'd give every one of us a key to the building. And this is a building full of like expensive equipment and stuff. So it was kind of like, it was just like suddenly being trusted with something.
Absolutely. And what age would you have been?
I don't know, whatever, what's the third year? Would that be about? Be probably about 20 or
20, yeah.
Like that. Yeah.
Amazing. Amazing that he did that. Like, is there something written on it there? 1B or something?
I think it just says 10. I think it's just a code
10.
for the key.
Okay.
So like the building's gone, but I still have the key.
That's gas that you kept it. And yeah. I think that's amazing. Being given the key like that at the age of 20 in your third year would kind of, you'd step up to the responsibility of it.
I think so, yeah. So I think that's something I kind of learned a bit, was to try and do that where I can. Like, I don't always succeed, but like, you know, when it's not assume that people who are starting out can't do things or can't be given responsibility. So yeah.
And where do you keep the key?
I don't know. It's just been ratting around at home. So heap of things.
Yeah.
I don't know the special.
Isn't it funny the way we keep these things? I think that's brilliant. And this other now little funny sketchy.
Oh, it's squiggles. Yeah.
You might tell us maybe what your name is and who you are.
Oh yeah. I'm Denis Shields. I'm based in the Conway Institute in University College Dublin, and I work in the field of what's called bioinformatics, and it's basically studying DNA and protein sequences on computers and looking for patterns. So
you're not in a lab with a pipette?
Yeah, no, not really. I started out when I was an undergrad. I tried doing lab work and I just didn't have the patience for it. You have to have a certain mentality to be good at lab work, and I wasn't really good at it. So I I ended up on computers, and so I do a lot of computer analysis. But what this came out of was there was a call for grant ideas about COVID, and like, you know, when COVID came out first, everyone was trying to think, oh, it's really not very nice. I should be doing something too. So I kind of said, well, what should I do? So I kind of thought, well, I know a bit about peptides, which are small bits of proteins. We'd done some experiments in the past in heart disease, trying to look at them. And so I thought, well, can I apply some of that, what I learned from that, to designing peptides against COVID? And I was saying, how am I going to do it? And I was thinking about different bits. But I can't even understand this picture now. Like, it makes no sense to me really. Well, it makes sort of sense. I think if I spent half an hour, I'd probably, but at the time it was useful for me to get my ideas straight, just scribbling all this down. So
you were kind of thinking immediately, and there's some kind of codes and different, you understand, if you looked at it again properly, you might be able to.
Look now, and I'm going to explain it all to you in a lot of detail. Yes, there's different, peptides. So there could be like little motifs in peptides. Like there's a famous motif on our blood cells, our platelet cells in our blood, called RGD, which is important for binding. It's on the fibrinogen, the clotting protein that binds to these platelet blood cells. So that's a very important motif, and I was interested, you know, could you build from known little short motifs? So RGD just means 3 different building blocks of the amino acids that make up protein.
Yeah.
And then there's other building blocks, and I was trying to think, well, what if we made combinations of these, put them together in ways that we could maybe make them only be activated when the virus is infecting the cells. So basically I thought about this and thought, well, I can do some computer analysis to try and come up with these things, and what would be interesting parts of the viral protein to introduce into these peptides along with other bits of peptides. And then I found collaborators in the protein chemistry, the peptide chemistry, people who can make these things, and in virology, so Virginie in virology, and Dave O'Connell in the protein biochemistry end of things, to help us make some more of these peptides. So we've kind of working together on that. So this initial squiggle diddly has led to a grant where we're actually just coming to the end now this summer of some of the work on that. So.
Okay, so when you say you identified these motifs, did you kind of just dream them up out of your head, or?
Yeah, I mean, a lot of them, I think, in retrospect, probably what we did in the grant, what I said I was going to do in the grant are a little bit different to what we probably ended up focusing on when you actually sit down and start doing things. But some of these were based on motifs that are in the virus sequence that we know bind, like there's a motif called RGD, it's actually in the coronavirus spike protein. Like, so that RGD, that was why it was interesting as well, apart from what I was saying about it being important in blood clotting cells. So, and then this one, the NRP1 binding motif, is just after a cleavage site on where the spike protein gets chopped off, it reveals this binding site for this protein called NRP1. So I thought, could we make something that combines some of these motifs together? Would that have interesting signaling properties? That was my original thought, and we're kind of doing that at the moment. I think we've shifted our direction a little bit away from those two motifs for a number of reasons, but the principles of the sort of the idea behind it are there.
Okay, so probably your background in genetics and has allowed you to really fully understand the genome, the code, and then be able to, like when you see the letters of the code, can you then sort of almost visualize the protein, or? Yeah,
sometimes it's easier than others. I mean, sometimes you can visualize little aspects of the protein, but not the whole thing. There is great software now that's just come out the last 2 years called AlphaFold that uses machine learning that actually works, that is very good at predicting from the sequence of letters the way that string of a protein kind of folds up into a three-dimensional thing, a particular shape. And so that there is good software out there that does that. But sometimes you're just I'm often just looking for simpler patterns, actually, to be honest. And sometimes it's things like, it's understanding the evolution of these things. So if we compare the SARS-CoV-2 with maybe the common cold coronavirus, and then you look at the spike protein and you say, what bits are different here? Does that give clues? Is that the bits that are the same, are they important, or are the bits that are different because that's what makes a difference between them? And, so
when you say you look at those two things, you mean you look at like reams and reams and reams of code?
You imagine you've got one protein sequence from the SARS-CoV-2 and one from the cold coronavirus. You just line them up against each other. And so you'll see a few changes where what happens is that the protein is inherited along with the DNA sequence from generation to generation. But as viruses evolve, there's mutation changes, which I think people are kind of familiar with now because you hear in the news about there's a new, I can't remember, but they give the actual letter, the position of the letter that's been mutated in the protein sequence and that. So I think that's really a lot of that's what I'm kind of looking at, essentially, looking at understanding those changes, maybe over a longer period than just within the human population in the last 2 years, because we've got the evolutionary history going way back where we compare all the viruses to each other, and then that gives us hints about function that might help us design peptides. Because there is a known peptide that's been used in HIV, Fuzeon it was called, and it's about 40 building blocks of amino acids that worked quite well to help people manage their HIV. And so the model of using peptides in viral disease is out there. It's been proven to be clinically effective. So it's worth a shot to have a think about these things. And if nothing else, we might learn something more about the fundamental biology of coronaviruses, which I think will be no harm for us.
Very. So are you saying that you've actually been able to make these peptides, and have you seen any kind of positive effects against the? Yeah,
we have seen some. So we've designed the peptides, we've synthesized them. Mark Devasel in chemistry in the College of Surgeons has made them for us with Niladri and Roy and Ajay Pal in my group, and they've been working together on the design and synthesis, and then in the virology lab, in Virginie's lab, they're testing. So so that we have tested them. Now there's nothing there that is, coming out as a real blockbuster. This is the must-have peptide that we really want to go after. But we're learning interesting things about some of these peptides, and we're looking to modify them then to build them to give them a greater sort of, like for example, we have a peptide that gives you 50% inhibition, but that's not much good, saying I've cured half your 50% of your coronavirus. You really want to do a bit better than that.
So it's amazing that the sequencing of the genome has allowed you to do all this, have all this knowledge and develop all this thinking on a computer by working on a computer all day long.
Yeah, it's amazing. Fun. It's kind of solving puzzles for me. It's like it's interesting.
Yeah. So this is an interesting thing as well. This is a piece of paper that I'm looking at here that's just covered in all kinds of numbers and letters and sequences. So just tell us briefly what that is.
Yeah, what this came out of was a few years ago I joined a music group that plays Indonesian music. So there's a very talented musician based here in UCD and also in the concert hall, Peter Moran, and he leads a group. And when I heard them play, I just thought it was the best thing I'd heard in a long time. So I ended up joining the group, and so we've played at a number of interesting venues, and it's a very interesting music because it's in a totally different parallel universe to Western music. You've got different scales and different structures and a whole different philosophy and mentality about the music. So it's just great fun. And then I was just sort of sitting at home one night, and I thought, oh, wouldn't it be really crazy to try and design a piece of music for this orchestra based on something from DNA or protein? And when I thought about it, I thought, well, the great thing about collagen is a protein which it has kind of triplet repeats in it. And I thought, well, triplet repeats sounds like music to me. So, and it also, like, then the DNA code itself is a triplet repeat. So you've got a triplet repeat within a triplet repeat, which is sort of like, I thought I'll have to play with that. So I looked for a chunk of collagen that looked as though it was repetitive enough and might have a bit of structure to it. So I just took a bit out of, there's a collagen 1A1, one of the big collagens, really important to our body. Like about 18% of our protein in our body is collagens, so they're an important group of proteins. So then I just tried to assign notes to certain amino acids. Now the scale only has 7 notes. Yeah. So and we have 20 amino acids, so I had to collapse it down a little bit, which is fine. I just, amino acids that are similar, I just gave the same notes.
I'd love to know what it's like to be inside your head. I have to say that you can do that, that you can.
It was just fun, so, and then we got to play it, and we played it in 1 or 2 different places, and it was good fun.
That's gas. And these objects here, this is some piece of a computer, obviously, which you wouldn't be able to do your work without.
Yes, yeah. Exactly. Yeah.
What is this actually?
That's a power pack for a computer, yeah. So that's just, it doesn't do any of the thinking. All it does is just passing electricity, and it's got a fan in it. And I don't really know a whole lot about how computers actually work. I've never actually taken one apart, but like most people, I use them. So
yeah. So I just put these on the table here. It seems to me that more so, I think, than in any of the other disciplines that I've learned a little bit about in science, it seems to me that what you do is really like looking for a needle in a haystack, or having a massive tangle and having to sort of root around and pull out one thing and see where does this lead or where does the other lead, and it's incredible to me that you are able to keep all of that in your head and have an understanding of the physical reality of it when you're looking all day long at codes and numbers and sequences, and I think it's really, really interesting. I've been, I'm Lorna Donlon. I've been speaking here to Denis. I was artist in residence in the Conway Institute last year and got to meet several scientists, and I'm really happy to be able to share some of the stories of their research and what they're doing. I hope you found this interesting to listen to.